All right, well, let's talk a little bit about Pokemon then, because I'm quite
Mm,
excited for this, because
okay.
I've never really looked into the sort of what the potential science around Pokemon could be, but you've given it a great deal of thought.
Yeah. I think about it a lot, maybe a little bit too much.
Yeah.
Um, well, unsurprisingly to anybody in one of my favorite types of Pokemon, because all the Pokemon have different types, so to speak, is grass Pokemon. And I am not going to go into the travesty of the fact that grass Pokemon are called grass Pokemon despite none of them being based on an actual grass species. That's okay. But I do want to talk about some of the interesting Pokemon that are inspired by plants and some of the really interesting science behind those plants and the biology of why they're so incredible. So, I may as well start with a Pokemon that you have gotten particularly excited about, which is Bulbasaur.
Yeah.
Bulbasaur is one of my favorite too, I'm not going to lie. It's a dinosaur with a plant on its back. How can you not like that? But Bulbasaur evolves eventually into a Pokemon called Venusaur. And its name, Venusaur, is a combination between the Venus flytrap and dinosaur. Ironically, it is not based on the Venus flytrap, it is based on the corpse flower. And
Mm.
a lot of people know about the corpse flower because it is the very, very smelly flower. Similar, well, it's sorry, not based on the corpse flower, based on the corpse lily.
Mm.
It smells very bad. It smells like a corpse, hence one of the reasons why it's been called that.
Hmm.
One of the really interesting things about this is that a lot of the time people wrongly associate it with being a carnivorous plant. And I'll talk about carnivorous plants later, but the corpse lily is not a carnivorous plant. It is a parasitic plant. It is endemic to Southeast Asia, and one of the reasons why it is such an interesting plant is that it doesn't grow leaves, stems, or roots. It is just a flower. And that, in and of itself, is bizarre. So, being a parasite, it grows itself inside other plants. It requires essentially the life essence of another plant in order to grow itself. It doesn't have leaves, so its photosynthesis is compromised, so it has to
Mm.
siphon the life from another flower. It's considered a parasite instead of having a symbiotic or like a beneficial symbiotic relationship because it could kill the plant, but it doesn't seem to always be the case. One of the reasons for this is because species within this genus of corpse lilies, vary so much in size. So they can go from about 12 centimeters, which is still a really big flower,
Mm.
to about 1 meter long in diameter.
So that's huge. I
So
mean, when it comes to flowers, that's really large, right?
it's gigantic. So even if you're looking at something like sunflower, which people consider a big flower, like that's got nothing on the corpse lily. So it is really interesting that for a plant that doesn't photosynthesize and doesn't generate its own nutrition like most plants do, that it can grow so big.
Hmm.
Um, one of some of the biggest flowers can weigh up to 10 kilograms, which is just insane.
Yeah, like that's really hefty. And like,
It's hefty.
I
Yeah.
guess like not having leaves. I mean, that would be like a human not being able to like have their own mitochondria. I guess like not having their own little energy
Mm-hmm.
factories, because
Yeah,
leaves have
exactly.
chloroplasts, right, which turn
Mm-hmm.
light
Yeah,
into energy via various processes. We have a similar
exactly.
thing, but we don't use light. We use like sugar and stuff. So,
Exactly. And so that's it. These corpse flowers aren't generating that sugar because they're not photosynthesizing. So they are siphoning those kind of nutrients, those sugars, and those proteins from other plants. Um, so it is like from an evolutionary perspective, it is quite bizarre because you would think normally that it's just easier to make your own protein when there's sun. But a lot of these plants, which are either parasitic or carnivorous and don't have systems, come from places that don't have great soil, which
mm,
is interesting because Southeast Asia usually does have quite good soil and a good environment. But these ones clearly benefited from not making it themselves. But that's probably why they can grow so big, because they're not expending energy growing those other organs. But plants still need nutrition, not just from something that they're using as a parasite. Not nutrition, sorry. Plants still require other outside influences. So most plants will require some form of insect interaction. So some plants will be pollinated via the wind. Some plants will be pollinated because pollen gets stuck on an insect. It goes to plants and pollinates the rest. The corpse flower is really, really smelly. So insects that like to eat dead animals and feast off that will often come over and they'll smell the corpse flower, drop in, pick up the pollen, and go away. This is, I think, pretty cool because usually we think of flowers as being nice smelling, and so it attracts the insects with the nectar, which means they are only attracting certain species of insects, just
like
the
bees or
ones
something,
more like
right?
flies. Yeah, so they aren't going to attract bees. They're more likely to attract things like flies, different types of flies. But that means the fly will come pick up the pollen. They're not going to go to any other plants. They don't care about sweet smelling smells. And they'll go to another corpse flower. So it's actually quite efficient in terms of pollinating each other.
It's like a great trick. It's like, instead of, like, imagine window shopping. And like most people, they want to window shop like, oh, there's a new phone. They've got some DVDs. Maybe I'll check out these clothes. But like this plant is like, you know what? I just want the people interested in model trains.
Yeah, exactly.
And there aren't many model train shops, so surely they're just gonna like go along to my shop or my friend's shop down the road. That's like an amazing kind of like innovation for a plant to kind of completely flip the normal, like the normal way that plants work.
So it's found its niche and it's really gotten in there. Completely unrelated, just because it reminded me of what you were saying about finding model trains and stuff. Figs, the species of fig is only pollinated by a single species of an insect. So that's another, they have found their niche and they are going for it.
So this is an interesting question for you then. Are figs vegan?
Okay, so this is an interesting question. So the fig is pollinated when usually a female wasp of some sort usually comes along and burrows into a fig and it will lay its eggs. They will be male and female eggs that hatch out. The females then mate with those males inside the fig and then try and escape and go and find another fig to lay their eggs. In that process, because there are male figs and female figs, eventually they'll land on a male fig and eventually go and find a female fig. So the pollen spreads between male and female figs. But because they have burrowed into a fig and out of a fig, female fig is only pollinated when another wasp comes in and burrows inside of it. So a lot of people, look at that and they're like, but that's not vegan, it means we're eating insects. The thing with that is that there are so many enzymes within the fig that usually if a female wasp has burrowed, or a male, it doesn't matter, usually the males don't make it out of the fig though. If a wasp has burrowed inside of a fig and died there and not had its babies, or had its babies and just died anyway, the enzymes inside the fig almost always completely dissolve the wasp. So there are very, very little traces of insect inside those figs. And so it's kind of like the whole idea of, well, all plants mostly are insect pollinated if there are no physical traces of the insect in there anyway, because you could land on any kind of flower and just die there, or drop, whatever, have a fight with someone else, your leg falls off. You're kind of getting into those areas of how vegan is vegan. Yeah.
No, I just think it's an interesting question because obviously, the way the fig reproduces is really interesting. And I would say figs are vegan friendly, but if you want
to... It's a difficult question. It's more that if you were eating, there is almost no trace of insect left.
Yeah.
usually.
Yeah. So that's the story of Venusaur. Are there any other like grass, like alleged like quote grass type Pokemon that are like have interesting kind of like backstories that kind of segue with your love of science?
So I do have one more. That's Victory Bell. So I love Victory Bell. It is just a giant big pitcher plant, essentially. And I remember watching the anime when I was a kid,
Yeah.
and James had a Victory Bell, and it would always like try and gobble him up. So it was kind of getting to the roots of carnivorous plants and what they're like. Pitcher plants are predominantly found in North America. We do have quite a lot of carnivorous plants that are native to Australia as well. Definitely go have a look at them, because there is one that has a sticky kind of leaf kind of structure, and when an insect lands on it, it catapults it into its belly. It's pretty amazing. So predominantly in North America. These parts of North America that most of the carnivorous plants grow don't have great soil. And that's a really important thing when we're coming to look at carnivorous plants. The quality of the soil is very, very strongly correlated with their evolution, understandably. And I'll go into why. So the insects are attracted to how the plant look, not based on things like the smell, like with the corpse flower or normal like roses or whatever. They look at it and they think that there's nectar inside, because it looks like it holds it. It looks like a cup, and you can kind of see the shimmeriness of the liquid on the outside. So they go in to have a look. They crawl down the side, and because the sides are very, very smooth, the insects can't grip on the side of it, and they literally just slide down right into the bottom of the pitcher, because there aren't any hairs. So some of them will have hairs on just above the surface of the digestive liquids inside them, so that when the insects try and get out, there's like a physical barrier keeping them inside. So it's actually super morbid, but amazing. So the liquid in the bottom of the pitcher plant drowns the insect.
Ooh.
And then there are, yeah, I know. So they don't actually die from, usually with the pitcher plants, they don't die from being digested alive. They'll drown. So I don't know which is better, but regardless, still weird. And then there are lots and lots of mechanisms that go into this pitcher plant, which helps the nutrients from the dissolved insect be consumed. Not only will there be things like proteins and enzymes, but there are a lot of bacteria that live in those digestive juices, and larvae as well, that live in those digestive juices, which help it all get gobbled up. Interestingly, not all species of pitcher plant eat insects, just insects. So there is the Nepenthes ioe, which eats shrew poo.
Huh.
And it's so bizarre. They are literally shape that the shrew come and poo in them, or accidentally or otherwise, and they absorb the nutrients from that. But I think the most interesting thing of all when it comes to carnivorous plants is, if we're looking at the evolutionary history of a plant that has involved carnivorousness, like how do you think they're related? Not a hypothetical question.
Right. I mean, you'd think that they would have some common ancestor that would sort of, like the idea would have sprung up, and then they would have somehow throughout the world kind of, they would be related in some way. But I'm guessing you're going to tell me that they are not.
Absolutely. So intuitively, it seems like share a common ancestor, but it seems like multiple species of carnivorous plant are actually a case of convergent evolution instead of divergent evolution. So they do not share common ancestors. The trait they share in common is that they have poor soil. But one of the reasons that this is possible, that they can evolve from all of these different plants completely separately and independently of each other, is because the proteins and enzymes that are en route that are responsible for absorbing nutrients through the soil and the water into the roots have not changed. Those proteins are still there, but they are now being expressed or switched on in different parts of the plants up near the digestive juices. So it's less of a result, it's less of a case of new genes evolved and more of a case of the genes that were already there were allocated to a different area of the plant. So all of them started with the same genes. And if you were looking at the genetic history of the plant, they share the common ancestors, like the gene common ancestors. But if you were looking as a species, they don't share a common ancestor.
So this is like
I think it's go on.
It's like the ultimate example of like the sort of the Darwin idea of how evolution works, right? Where you have an organism and it finds itself in a certain environment, and in order to survive, it just has to change over time. And at some point, the funny thing is that the way genes work is that for every gene there's almost another gene telling it when and where it should be switched on, and clearly one day for whatever reason some switch got flipped somewhere and the gene just ended up somewhere else, and all of a sudden this plant can just survive where other plants couldn't, which is crazy. And this has happened multiple times, you're saying?
It's happened in most carnivorous plants that we see. They have come from different ancestors. Just the same genes have been relocated, essentially, not relocated, relocate, but the proteins and the expression of the RNA has been relocated. And I think also this is a really interesting way of highlighting why we look at phylogenetics and the evolutionary history of not only a genome as a whole, but individual genes.
Yeah, it's interesting, right? Because sometimes the whole genome actually tells you less of a story than a few key genes, because a lot of things are actually pretty well designed from a biology point of view. Like evolution over time has engineered really elegant solutions. And so it takes very extreme changes to actually drive evolution to happen. And so
absolutely.
And so for me, that's actually really interesting as well, how sometimes really small parts of the genome can actually tell you a much bigger story than the genome as a whole.
Because if you're looking at the genome as a whole, convergently evolving instead of divergently evolving, it does look very complex. But when you were looking solely at that single family of genes, and there's a few other families as well, it's actually really simple and very elegant, as you said. I think the other thing when we're looking at Venus as well is the difference between what is science and what people perceive as plants, and their scientific role and their evolutionary. Because you look at Venusaur, and Venusaur is clearly based on a corpse lily in terms of design, but they have gone ahead with the nomenclature of naming it after a carnivorous plant. So clearly there is a dissociation between what is a parasitic plant and what is a carnivorous plant, but they're still leaning into those themes and ideas. And whether that was deliberate or not, I'll never know. But it's always just been I hear all my friends talking about Venusaur as a plant. And then I could be like, oops, nope, I'll out-nerd you there. It's not. Just a parasite.
Yeah. Like, a really fascinating one, though. I mean, like, parasites are really interesting in and of themselves. Like, being someone who looks at infectious disease, I think a lot about parasites that infect animals, which tend to be like these little cellular animals or like worms and stuff. But, plants have their own completely crazy life cycle of things that parasitize them, whether it's insects or just other plants.
And this is actually an area that would both connect on, I suppose. Looking at the corpse flower and looking at the corpse flower is an extra interesting organism because it's not a virus, but it does horizontally transfer its genes. And so it can transfer up to, of its host, so it's on a host, of the host, like trees and things that the corpse flower is growing on, we have found up to 2% of their genome has been from the corpse flower.
So that's really interesting, because obviously when we talk about things like horizontal gene transfer, we're like literally talking about genes going from one species to another, rather than being passed on with like the children.
Exactly.
Which is normally quite a rare event. It's one of the ways that we think antibiotic resistance is happening. Bacteria kind of sharing DNA between other species. But I think in plants it's actually a bit more common than you might think. I seem to remember a story where the sweet potato, the reason that exists is because of a horizontal gene transfer event.
Interesting. I'll have to go learn about that.
Yeah, and they think that possibly a bacteria actually took up genes from one plant and then sort of somehow managed to transfer them to a different plant. Yeah, which is kind of crazy.
Plants are probably more susceptible to horizontal gene transfer and other crazy things mutation-wise because their genomes do seem to be strangely a lot more stable than animals and humans and things like that. And so there's a really great example I like to use with my students of watermelons. Right. Watermelons are diploid, so they have one pair of chromosomes, 2 chromosomes. But through completely reasons nobody knows, some watermelons completely duplicated their entire genome to end up with four chromosomes instead of only 2 pairs. They have 4, and breeding those 4 chromosome watermelons with the 2 chromosome watermelons ends up with a progeny that has 3 chromosomes, and that makes really inefficient, unbalanced meiosis, and so the seeds can't get fertilized, and that's why we have seedless watermelons.
Right. That's crazy.
Just freak evolution. They're like, yeah, I'll spontaneously duplicate. That sounds normal. Let's go for it. Off I go. Live my best life.
And it's quite common in plants, right? Because like strawberries have, I think, 6, and bananas have 8 copies of their chromosomes.
6. Makes it a lot harder to research. Makes it really difficult to research some of those really big genomes.
Right, because you don't know which of the 6 that you're even looking at, right?
No,
when it's just raw DNA.
So even if you're looking at grasses and crops, our most common model organism when it comes to grass crops is rice. And rice has about 470 million nucleotides in its genome, but wheat has 17 billion. And
right.
No, but I don't want to siphon through all of that, but it makes the bioinformatics a lot more difficult. It makes the functional genetics a lot more difficult. And it's really the main reason why we even have model organisms in the first place. Because the more chromosomes, the more genes, the larger the genome, the more complex it is. Not complex, that's not really the right answer. The more complicated it is to be able to ascertain the function of those genes. And when we're looking at something like wheat, which is such a globally important grain, you've got to start something. Give me 470 million any day.
Yeah. Yeah, I mean, I work sometimes with like bacterial genomes, which are just like a couple of million base pairs, and they're quite easy to sort of assemble into, one continuous genome that you can look at. Yeah. But I can't imagine having several chromosomes and massive chromosomes and trying to figure out, if the gene is the same on each chromosome, or if there's a different version of the gene, or if the gene is actually twice on the same chromosome, just kind of separated.
All the time.
Yeah.
And so I think that's really also down to that difference between complexity and genome size and complicatedness, because I think intuitively we like to think of large genomes, lots of genes as more complex, but that's just not the case, because it's definitely not more complex than a lot of bacterial things that we'll find, or a lot of viruses. Genome size doesn't correlate to complexity. Yeah, it just makes a lot of headaches for us as scientists.
Yeah, pretty much. All right, well, I think let's move on a little bit to... I think given that we're already talking about sort of the way we communicate science, do you remember a while ago, after the US election in 2016, there was a real public outcry about the way information is presented on things like Facebook and social media? Yeah, I remember that.
So
after that, there was a bit of an initiative, I guess, to try and curb the amount of, I guess fake news is one of the terms that came out of that. Although that phrase itself kind of got a little bit co-opted in a lot of ways. At the start, fake news very specifically referred to completely fabricated stories that were literally just there to get views. You know, the actual content was completely made up. But then the idea of fake news kind of morphed into like stories that could have been earnestly written by someone but were wrong. You know,
So
like,
fake news has become fake news itself in a strange roundabout way.
yeah, weirdly. And so a lot of people wanted something done about this, and Facebook instituted this sort of idea of fact checking. And I don't know if you've ever seen it, but occasionally you will see a news article on Facebook, and a particular group called Snopes, who had a long history of sort of fact checking a lot of things, and a few other services. If you see something that is like patently incorrect, sometimes the Facebook team will have tagged the story with a counter article saying, you know, this article is likely incorrect, and this is kind of a place where you can get better information, which on the surface sounds good, right? And I think
it
probably
sounds like peer review, but for journals or
Yes.
for like general media.
yeah,
But
just,
who is the one who are saying placing these verified tags on them?
so Facebook has like a fact-checking team, and there are certain organizations which have a history of, I guess, like mostly like correctness, if you like. And obviously, judging what is correct is maybe somewhat problematic in and of itself. But sometimes things are unambiguous enough where you can say, well, this information just doesn't, you know, that isn't what happened. Like the events you're describing here, they just completely are not in line with like actual footage or eyewitnesses or, you know, more reliable sources.
It's just clearly not right.
Exactly.
Yeah.
And obviously a lot of the time with like medical stuff, you know, a lot of these like say cancer cures or things like that, that are essentially like pseudoscience. These organizations have a long history of sort of fact-checking those and trying to research the real science and seeing if there are actual studies and things like that.
Yeah.
So in some ways, these warnings have been good in that they have definitely helped on the specific links that they're attached to, where people will see them and then they will think twice about whether that link is sort of actually correct information or not. But it turns out this initiative has somewhat backfired in another way. And can you maybe guess what way that might be? There's a few ways, but
there's a couple of things that I could think of. So one of them being that whoever choosing the verified and unverified could put misinformation on it and then say true or false in the wrong situations. But then also, I feel like if it's just saying verified, or because unverified doesn't mean it's not right, but verified means it's probably right. That like right is not always the right term, ironically.
Right. Most correct is sometimes better.
Most correct, but not even the most correct. So if we are looking fully from a point of view, and this is going to be the same for a lot of things. If I'm writing a paper, I'm not right about something. I'm just saying all the evidence to this. But that's different from being right.
Yeah. Yeah. It's like this is the most likely explanation based on the evidence we have now.
Yes.
But if that evidence changes, then maybe we have to change the way we're thinking.
Yeah, exactly. And so if you are saying something like, this is true, or this is not true, I feel like people would jump on those labels and kind of misuse them.
So that's definitely one thing that has happened. In fact, some of the sources that were originally on tap to kind of be the arbiters of fake news, one of them was Breitbart, and I don't know if you've heard of them, but they're an extremely right-wing media organization, and so that was problematic. But what these researchers found actually, and I kind of like this, is they've called it the implied truth effect, is that one of the things that was happening is if people, because Facebook can't get to every single article, if people didn't see the fact check tag on a story, They just assumed that the story they were about to read was true, even though in reality, a lot of the time what happened was it just hadn't been tagged with anything at all. You know, it had neither been tagged as being verified or unverified. And what that means is that a lot of people are seeing pseudoscience that just hasn't been fact-checked yet. But because it doesn't have that tag saying that it's wrong, they just assumed it was correct.
Interesting. So it was like the opposite, because I would have expected, okay, let's be skeptical of anything that's unverified. But they were doing the opposite. Let's accept anything that hasn't been unverified.
Yeah, so because they got used to seeing these tags, if something didn't have any tag, they just assumed that that meant it was fine because it hadn't been tagged with like a pseudoscience or fact-check tag, which is...
Wow.
It's kind of a problem, right? Like,
it's a major problem. Yeah.
I think one of the things is that what this fact-check does is it doesn't incentivize you as a person to think critically for yourself. A really great example... Because you're allowing
other people to do it for you.
Exactly. And that means that when you actually don't have someone to do that for you, you still don't have the tools to look at something critically and decide for yourself if you think it's likely to be true or not. I remember this example, and this is one of the very low stakes places where this doesn't matter that much. But I remember once a bunch of my friends had shared this competition to win Qantas flights, and I remember seeing the post and thinking, wow, that image doesn't look as professional as I would expect from Qantas. So I had a look at the page, and it turned out the page only had like 3,000 or 4,000 likes. And the name of the page was Qantas with a full stop instead of just Qantas. Yeah. And obviously it turned out this was not a real Qantas page. The real Qantas page has 100,000 likes. Obviously, Qantas is a trademark name that they would have like ownership of on Facebook. Yeah. And if people had looked at the post for themselves, they could have found out the same thing. Now, imagine if you were used to seeing a tag that when you saw those kind of things, there was a warning coming up saying, this is fake. But then a new one pops up that tag isn't on yet. You might just assume that this one is fine because it doesn't have the tag. So I think one of the unfortunate things about doing the fact-checking on behalf of people is that it's good in that, like, most people don't have the ability to fact-check everything. There are plenty of stories that I would come across. I would not have any of the skills or knowledge to know whether it was real or not. But obviously, what we do want to teach people is the general things, the general markers of where, like, there are certain signs a lot of the time that things might not be true, certain language people use, the lack of certain language sometimes, or the lack of certain features. And I think that's really what we should be trying to do on Facebook, is try and give people not just necessarily like, here's the fact check, this isn't true, here's the real story. Actually go through why the story is fake, and maybe people might next time be able to actually see for themselves when they read something, oh, that's flagging in my mind that reminds me of that other fake story that I saw, and and that's the reason it was fake.
Exactly, and I think it comes into when we're looking at skepticism in general, and we like to say skeptical as scientists, and that doesn't mean we don't believe things, and we're trying not to believe things. It just means we're trying through that critical mindset, through that critical microscope, essentially, to be able to say, hold up, that thing has a full stop at the end of it that looks a bit strange. Yeah. Maybe there's something going on there. And I want to say that, you know, make sure you follow scientists on your social media, have that knowledge, and those words will become part of your everyday way of seeing the world, and you will learn those skills and stuff. But you know what? There are scientists that have their own agendas as well. And there is nothing wrong with them being skeptical towards that. But if we're looking at, well, who are the experts, who's saying verified, who's saying unverified, the same thing can come into that. Yeah. And you still need to analyze all of that. And it's part of looking at scientific consensus and things like that. Well, most of the scientists believe this is the case, or whatever like that. And kind of analyzing that and saying, yeah, you know, there's a reason for that. I can see why they believe that. I can see why most scientists think climate change is a real thing. Right, because of this and this and this.
And
Not
even,
just, I believe it because they think it. I
yeah,
believe it because I have now gone and seen the reasons why.
exactly. And I think as well, it actually extends beyond science, that a lot of political stories come out and sometimes, you see it and you're like, hang on, is the story I'm being told here really representative of like what these events actually mean? Like it happens in the way the government, and not just the government, companies, people, anyone with an agenda, the way they frame economic figures, the way they
Yeah.
frame things like job growth, for instance. If people say, well, unemployment dropped by 60%, but unemployment was say only
And it's because
2%.
everyone died.
Right, or yeah, exactly. Like sometimes there are just reasons why figures look impressive, but they're not. I think,
Yeah.
obviously, to go back to science, one of the ones you see a lot is, eating bacon every day increases your cancer risk by 1,000%. And it's
Yes.
like, okay, that's interesting, but that doesn't mean you have 1,000% chance to get cancer now.
No,
If the chance of
exactly.
getting cancer before eating bacon was 0.0006%, 1,000%
It's,
increase is only like 0.006% now, which is still not very
yeah.
high. And
And this is when we look at different stories coming out, especially in media, saying scientists have found that lettuce cures cancer or something like that, and they're like, well, actually, a particular protein from lettuce helped with one particular cell type of, you know, in one specific situation in mice,
yeah,
and that's a
exactly.
completely different thing.
Yeah.
But that can then go and I'm guessing be unverified, in which case people are like, no, those, lettuces are relevant.
Yeah.
But so it can do both. I really found something interesting recently where I people discussing not believing science and the consensus around science due to scientists changing their mind, which I don't like wording of that, but
Right.
essentially science is always changing, it's always growing, we are always refining our methods, we are always refining our hypotheses and things like that. And people looking at that and saying, well, you know what, they were wrong about this thing 10 years ago, so why should I
Yeah.
believe them now?
Yeah.
And it's worrying that people look at that because I'm much more inclined to, so I'm wrong, they've probably done a lot more research.
Exactly.
But it's kind of reminding me of this as well, of jumping on that unverified stuff.
Yeah. Yeah.
Don't know why you'd want to, but that's what people do, I guess. And that's one of the reasons why we need that kind of consistent science communication and teaching people how to use those skills. Because if you can't read a scientific paper, that is absolutely understandable, and it
Yeah.
creates those problems of lack of science literacy in general. But
Yeah.
if you can't read a journal article and be like, okay, that really doesn't sound right, that means we need to work on other skills.
Yeah, exactly. And I think it's one of these cases as well where, it's the job of science communicators and journalists to sort of bridge that gap a little bit, but the resources to do this are just decreasing all the time, essentially.
Absolutely.
And unfortunately, so like this show is a completely amateur hobby production, right? No one's paying me any money to do this, but
No.
it's kind of a passion of mine, and
I just think it's important.
yeah, it comes down to people with passions these days to kind of get these messages across. And I think it would be great for there to be more researchers, and not just for science research, just for really solid journalism in general, for a lot of things. Like there's a lot of things happening in the world that could do with a bit more scrutiny from,
Yeah.
from back in the day. There would be journalists working around the clock trying to get the scoop and trying to get the inside information, and not just because they wanted to sell papers, because they cared about the truth.
Yeah, exactly. And when we're looking at journalism, whistleblowing, and investigation, that was always one of the things I remember when you watch movies and read books and all of that kind of thing, being like, yeah, the journalists are the ones out to find the truth.
Yeah.
And that's not always the case. And
No.
it sucks that we have to be the ones as the general audience to then look at that and review whether we truly believe what they're saying.
Yeah.
But that's also just what's happening right now.
Yeah, unfortunately. Well, let's move on from that slightly depressing topic. But I mean, the thing is, actually, before we do move on, it is important to know these things, though, because we need to know how to move forward, I think. And that's why research into these things is really important.
I don't think that the people who are sharing the pages that haven't been verified either way and checked, I don't think they're doing it to be bad. They're doing
not.
it because they know that there's false and there's not false. And so they're deliberately trying to not share the things that are false or haven't been verified to be false.
Yeah.
They're still trying to make those right choices. It just means that there is a disconnect between how to make those right choices.
Yeah,
but
absolutely.
I think it's great that then people are making the conscious decision to say, no, we reject that journalism. We reject those particular articles because they aren't right.
Exactly.
That's great.
Yeah. Well, I wanted to talk about this because it's kind of interesting to me. I don't think it's too exuberant to say that like finding the existence of life off of our own planet would be possibly the greatest scientific discovery of all time. Not necessarily the most impactful, interestingly, because obviously things like vaccines have been amazing and public health measures and social change, all of those things have been really important for our like advancement as a society.
Absolutely, but there's nothing that captures the imagination more than thinking about extraterrestrial life in terms of science.
Exactly. I think just the idea that there is another, not even society, just that life is there somewhere outside of our own planet. It's so exciting, and I would really hope that knowledge would bring humanity together a bit to kind of realize like, whoa, like we're just a small part of this massive universe, and that maybe we need to look after each other a bit more. So in saying that, obviously I'm not here to tell you that we've found evidence of life off of the planet. I feel like you would already know if that was the case watching this. But I do want to talk about a very curious incident where, well, not even an incident, just a new discovery, where some scientists have looked at a meteorite that fell to Earth 30 years ago. So this meteorite's been looked at before, but one of the cool things about, I guess, science, and I think highlights the importance of places like museums who hold these big collections. As technology advances, we can look back at things that we had in the past with kind of a new lens and see if we can find something else. And so I guess the sort of the take-home message from this is that these scientists think that they've actually found a protein on this meteorite, and that the protein itself is actually not from the Earth. So they don't think it's contamination, and I'll go into why that is a bit later, but they have some good reasons to think that. We'll see if that holds up to other people's scrutiny, right? And I think this is this really ties into what we were talking about before when it comes to looking at a paper, deciding how credible it is, and seeing if other people can replicate the work later in that sort of peer review process. Because currently it is not peer reviewed. So I'm sure in your job you would know about arXiv. The sort of it's like a preprint server. So it's essentially where researchers will put papers that they haven't put into peer review yet, but they would still like feedback on, or they would like the information to be out there, kind of thing.
Yes. Yeah.
So this has been published in there, and I want to say that up front because sometimes there are science sites that kind of don't mention exactly what it means to print on bioRxiv, and they talk about these things as like definitive discoveries.
Whereas it's definitely not.
Yeah, exactly.
That's not to say that it's going to be completely wrong or anything like that. It's more just that they haven't been reviewed. They haven't gone under that scrutiny. They're still early stages.
Exactly.
So draft, essentially.
So it could be that in like 6 months' time, we're talking about this story again, but in a way that we're saying, yeah, it turned out this was a flaw, or this didn't actually turn out to be a good way of looking at it. And I actually think it's one of the things I really want to bring to the show is that we're not just going to abandon topics. If we talk about something and then there's a development, those are the stories that don't often get as much air, but I would love to talk about, the process worked and we looked at this and it turns out probably wasn't true after all. And I think it's important for people to know that as well. But so the thing is that there are certain things that we know are required for life. Weirdly, cyanide is one of them. which is actually kind of an important building block in life. But there are other kind of more like things you would think of, like amino acids, which are the like individual building blocks of proteins, certain organic compounds, and of course ribose, which is the sugar that's kind of like the backbone of RNA, which is the single-stranded version of DNA, a slightly different version that is used to sort of go from the instruction manual to the factory floor, if you want to kind of put it into analogy. And the cool thing is that these researchers have gone back to some of these meteorites that had some of those things. And I guess the reason is it makes sense that if it had the building blocks, it may have some of the actual proteins or even actual organisms, if they were super lucky.
Yeah.
And so they did this. And the thing that they have now that they didn't have before is this like a superconductor X-ray source. And so it's basically a really advanced type of mass spectrometry. So have you used mass spectrometry a lot?
I have. Not a lot. I would definitely say not a lot, but I have used it, yes.
So like the way mass specs work for people in the audience is that you essentially fire molecules at a sensor, and depending how long the molecule takes to get to the sensor, you can kind of tell how heavy it is. And if you have a bunch of different weights, you can figure out what the actual molecule is made of by comparing it to like molecules that you already knew, and you put through the machine. So now what they found is a protein, essentially. And obviously the protein itself doesn't necessarily mean that there is extraterrestrial life. We've known for a long time that amino acids and ribose and these things just exist in the universe. In fact, it turns out amino acids are really abundant in nebula. So amino acids are out there. But what we haven't seen before, we haven't detected, is an actual bunch of amino acids that assembled themselves into a protein. Now, obviously, on Earth, the major source of proteins is life. You know, life is essentially DNA that found a way to turn itself into proteins, to turn itself back into DNA, into proteins again, and you know, and so on and so forth.
Cycle.
Yeah, exactly. So it's kind of really interesting that they're looking at this. Now, obviously, I think a question that most people would have is, how do we know that this protein wasn't a contaminant? Because proteins are everywhere on Earth. They're really hard to get away from. You know, just your fingerprint leaves behind a whole bunch of proteins. And basically, what they did is they found previously these glycine amino acids, and glycine is one of the most common amino acids in the body and on Earth. And they found that they had like a stronger signal than in analyses that were done before. And what they actually found as well, though, which is interesting, is that the glycine was actually bound to other elements like iron and lithium.
Right.
And essentially they could tell from that rather than this just being little pieces of glycine, it was actually part of a protein, because amino acids have these functional groups, and obviously the amino acids, they all assemble into these biggest like 2D structures, and then the 2D structures then turn into these bigger 3D structures that we think of as proteins. So they called the protein hemolymphin, which I guess is because it's iron, lithium, and it's a protein. You know, heme being iron, lith being lithium. And it actually does have a structure similar to some terrestrial proteins, which again makes you think, oh, does that mean that this is contamination? So here's the sort of, I'm not going to say smoking gun, but here's the evidence that is at least pushing you in the direction of thinking that this might actually not be from the Earth, but be from space. So basically the ratio of deuterium to hydrogen, that's, you know, hydrogen is a single hydrogen, whereas deuterium, I believe, is a hydrogen with 2 neutrons. Which is a heavier form of hydrogen that is more like, it's more common in space where there's a lot more energy. Correct me if I'm wrong in the comments, but I believe deuterium is the 2 neutron version, of hydrogen. And basically, because of that, the fact that it had all this deuterium, proteins on Earth wouldn't have that ratio. However, hydrogen that comes from places like comets or like the very early proto-solar dust, like the stuff that our solar system is made of, that would be more likely to have that kind of deuterium-rich hydrogen. And that's
Okay.
why, potentially, these are proteins from outside of our own planet, which is really cool.
Wow. So was this a folded protein?
I don't know if they could get resolution enough to know if it was properly
Okay.
folded. I think they got some of the structure. I think they would have to do more advanced X-ray or like cryo-electron microscopy is quite popular now for doing that.
But presumably now that they've seen that there might be this potential, yeah, they can start kind of experiment.
Yeah, so I think that's the plan. That what they want to do is they want to take these proteins and try and model them and see if they can replicate the structures or basically take what they think the structure is and then they can get information on that and then see if the meteorite information matches that still. What they want to determine is whether they're actually looking at a protein which has that, as you said, that kind of folding, and that's
Yeah.
what makes the structure. Or they're saying that another possibility is what they're actually seeing is a polymer, which is more just repeats of short structures that
Yeah.
would not necessarily be a protein in that it wouldn't have that folding and that sort of
Yeah.
that structure. So I think
Because this is amazing because even just trying to find DNA in the universe is such a big thing. But protein itself, because you have DNA, which is transcribed into RNA, which is then translated into amino acids, which fold into a protein, that's still a lot of processes to get to a protein. And then you
Yeah,
have to go
exactly.
through the whole, well, is it a functional protein or is it just some
Right.
random pieces of amino acid that doesn't do anything? And then if we're
Exactly.
looking at functional proteins, even in terms of the genome, it's a relatively small percent. I mean, it's a small
Absolutely.
percent that we know of. So even looking at the human genome, I think it's about 2% of the human genome actually codes for protein coding
Yeah, it's very small
proteins.
amount. Yeah.
Yeah, so it's like infinitesimal chance that we can find these things. And I think that's what fascinates me so much about this, because
Yeah,
it's even
exactly.
smaller of a chance than just finding a bit of DNA.
Yeah, or at least like the building blocks of DNA, right? Like the sort of the sugars and things. So one
Yeah,
of the
exactly.
interesting things is that there was actually a study done on the International Space Station recently. And it turns out apparently in the reduced gravity of the International Space Station, it's actually easier to make protein than it is here on Earth.
Oh,
So molecules that would be unstable on Earth are actually they can build these large proteins and then bring them back to Earth once they're assembled. So
wow.
these proteins could never be assembled on Earth, but once they are assembled, they have this really solid-like structure, and then they can be brought to
Right.
Earth. So maybe in the reduced gravity of space, proteins are more likely to just kind of fold themselves.
Yeah. So it is quite possible that it is literally just formative gravity around it that can allow it to become a structure that doesn't do anything. But I
Yeah.
hope that it does. I think
Yeah.
that it would be like it would easily become the science of popular culture generation if it
Yeah.
were found to be a potential functional protein.
Oh, absolutely. It would literally be one of the most important stories in like a decade, essentially. So,
Exactly.
but yeah, we'll be keeping tabs on this. If it turns out that new evidence comes to light or people actually do some more analysis and we find out more, then we'll definitely come back and see how things are progressing. Well, that is all the time we have for today. Debbie, thank you so much for coming on to the show. Now,
You're welcome.
obviously,
It's always fun to talk science.
yeah, and especially if I can mix that science with some nerdy popular culture, then that is absolutely the best situation for me.
Absolutely.
So, look, I mean, you have a lot of like little ventures going on, and hopefully soon to be big ventures. So why don't you just tell
really
people you have like you're on Twitter, you're active on Twitter. Obviously, you have your company. So why don't you tell people how they can find out about all the stuff that you do?
Yeah, so easiest way to find me is on Twitter. My tag is at Dr. Remu. It was literally just because I used to have a Pokemon podcast, and all of the professors in Pokemon are named after trees. So I named myself after a New Zealand tree, Rimu, because that's where I'm from. So Dr. Rimu, just DRRIMU, is how you should find me. And then my company is called Siren, SCIREN. So if you look up either of those things, drop me a message or tweet me, and I can do my absolute best to try and help you with all your science translation needs.
Yeah, awesome. Thank you so much. And that's it from me as well. Obviously, remember to follow us on Twitter, Science Club Show. If you search Science Club Network on YouTube, you'll find us, and also on Facebook. We actually have daily content that is coming out, and there'll be a daily content coming out tomorrow about the weird shape of our galaxy, also featuring Debbie. So check out that as well. Thank you for tuning in, and until next time, goodbye.